welcome to Innovation Celebration, the show where we celebrate recent advancements in science and technology, the people and ideas that make them possible, and the ways in which they aid human flourishing. I'm Angelica Werth.
And I'm Thomas Walker, and I wanted to start us off with a discovery of a new prehistoric species of human in Israel. This new species is called Nesher Ramla Homo. I got that right from memory. I'm quite pleased with myself for that. And so this is a really interesting discovery in a number of ways, because firstly, it fills in one of the so-called missing links in evolutionary history. And this particular missing link arises from the fact that Neanderthals, which are a completely separate species to Homo sapiens, our species, have human Homo sapien genetics in them. Mm-hmm. And there's been studies as well about to what extent Neanderthal DNA is still kicking around in modern day humans. And so, those are two separate species, but they seem to have genetic, elements of each other in them. Mm-hmm. So there's a question as to why that happened. Interbreeding is one possibility, but this discovery indicates that perhaps there is a common ancestor between the two. Well, there is a common ancestor, both primates, but there's a more recent, more direct, common ancestor between the two species that helps explain some of that lineage. Mm-hmm. And also the fact it's discovered in Israel is really interesting, because the Neanderthals are seen as being a European species. They're kind of adapted to the cold climates in Northern Europe during the Ice Age. Mm-hmm. And this actually seems to suggest that both species descended from a species, the Nesher Ramla, the dragon man, I think you told me.
Dragon man, I've seen it called, yeah.
They both descended from this common ancestor in the Middle East, and
or at least that ancestor was present in the Middle East.
Yeah, but there's Homo sapiens populations and Homo erectus populations in Africa and Asia as well around this time. So if the Neanderthals might have been a sort of European last part of a larger species group, there seems to be evidence of. Mm.
Which brings me to the question of what is the dating on this particular find?
Yeah, so this particular skeleton that's been found is about 120 to 140,000 years old. But the estimation is that the species itself probably goes back to about 470,000 years and probably came forward around to the time of this discovery, probably not much more recent. But that means it predates Homo sapiens as a distinct species. But overlaps somewhat. So, the human evolution and the evolution of any species is not, a species changes into another one and it's just, there's a period where both exist. A new species emerges, but the old one is still around for a long time. Homo erectus, Homo sapiens, and Homo neanderthalis, Neanderthals were all around at the same time, and now it looks like there was this species in play at the same time as well. Mm-hmm. So, sorry, were you going to ask something there?
Well, I was just going to ask, how do they know that this one is a different species?
So it's really to do with the skull structure. It's called the dragon man because it has this strange sort of collapsed looking skull with no chin and these really big teeth. It has a couple of skull features that tie it to the Neanderthals. I was going to come to this later, but I suppose I can come on to it now. The man who made this discovery, whose name I will read off the screen, is Israel Hershkovitz at Tel Aviv University. He was talking about the methodology, and he was saying that what he describes as paradigms shouldn't be applied. So what he means by that is that we have a tendency to apply our pre-existing models to new evidence. And so when this discovery was made, there were a lot of researchers trying to group it into one of the existing species, such as Homo erectus, Homo sapiens, or the Neanderthals. And he was being critical of that and saying the evidence from the shape of the skull is that this is a wholly separate species. So rather than trying to apply our existing models and force it into one of them, we need to recognize what the evidence is telling us and just say that this is pointing to a completely new species. That's probably what it is.
And that's a really important idea to apply to all kinds of areas. You don't want to let your worldview color what you're seeing. You should have what you're seeing be shaping your worldview.
Exactly. So this is a matter of what we call epistemology in philosophy. So it's the study of how we know what we know. Yeah. And there's a couple of different approaches to that. And the proper approach is the approach based on reality, which is where you look at reality around you, you draw conclusions based on that. But science being science, we don't know everything at once.
Right.
So you're going to have a gappy understanding of things to begin with, and as you can build a model, build an understanding based on that sort of partial evidence. But the more you get, the more accurate picture you can build, and you have to adjust that model as you go.
Right.
So this applies to paleontology, it applies to archaeology, it applies to astronomy, physics, just every area of endeavor, not just science, but it applies to our lives. Trying to figure out the relationship you have with someone. Don't decide they hate you and everything they do fits into that preconception. You want to, adjust it depending on what the evidence actually tells you.
Right.
So, this stands at odds with the kind of epistemology that most of human history has been guided by, which is a sort of religious epistemology where you have, the words in the book are fact, and then one of two approaches: either new evidence has to be contorted to fit with the fact, which is the sort of what we call compatibilist religion, where you accept science but then you kind of twist it in to fit with the faith, or you take the sort of what do you call it, sort of almost extremist evangelist approach, which is you just disregard evidence altogether. It's just wrong, or it's and those are obviously both flawed approaches. One is kind of worse than the other, but they're both horribly wrong. It's the evidence should be guiding you. And so what Israel was saying is that in this particular case, take the evidence at face value. Don't try and fit it into an existing model. If the evidence doesn't fit with your model, then you need to adjust your model.
Right. So did you want to just say a few words about how this, the importance of this discovery?
Yeah, so I was actually going to read off a little quote from Israel himself, which I will need to just pull up for a second. So he says that this discovery enables us to make new sense of previously found human fossils, add another piece of the puzzle add another piece to the puzzle of human evolution, and understand the migrations of humans in the old world. They went on to say, even though they lived so long ago in the late to middle Pleistocene period, which is 474 to 130,000 years ago, roughly, the Nesher Ramla people can tell us a fascinating tale and reveal a great deal about their descendants, which means us and the Neanderthals, basically, evolution and way of life. So that gives you some idea of the significance in a paleontological context. But the reason this is more significant, to my mind, is that it's, as I was just saying, it's filling in the gaps in our true evidence-based understanding of our origins. So it's more replacing our not just our stories from antiquity, but our more recent sort of muddled together conceptions of our origins with the actual true story of how we came to be. It's knowing ourselves and our place in the universe. So
which is very cool.
Yeah, and the closer we get to that reality, and then this is the thing, the universe is phenomenally complex, Earth and beyond, and we're always going to be just shining a light and picking up glints and figuring things out from that. And the more of that we get, and the more complete the picture we get, the more awesome it is, to my mind. So that's just wonderful to see those gaps being filled in. Make sure I've not. Oh yeah, another thing I was going to say is that this new species had stone tool making capabilities. So they weren't, a sort of ape swinging from the trees or walking on two legs for the first time. These were stone tool using, manufacturing animals who were sort of at a similar level of development to the Homo sapiens of the time. There's evidence possibly they may have interbred with both the Homo sapien and Neanderthal population. So these species they're species, but they're very similar. There's not really a comparable situation now with humans. It's more like sort of dogs and wolves, and
you see it with other animals, but they're very similar, but they're distinct species.
Yeah, exactly, and that's common throughout the animal kingdom. But oddly, we're kind of this outlier species that's not really got anything that's closely related to it. I mean, we are closely related to chimps, but we're much further from them than these species were from each other, if that makes sense.
No, definitely.
So I think that's all I wanted to say on this. And then you were going to talk to us about genetic engineering.
Yes. So it's a really fun topic, in my opinion. The particular development that I want to talk about today is a new advancement in CRISPR technology called CRISPR Act 3.0.
So what is CRISPR technology?
Well, we're going to back up a little bit first and try to explain. We're going to have 3 sections to this bit. I'm going to explain firstly what gene editing in plants does, what the purpose of it is, and the effects that it's had so far, and then what CRISPR technology is, and then what this new development is.
Right.
So kind of a 3-part thing here, but it's all related to human flourishing. So, and science.
Yeah.
So the first thing that I said, gene editing in plants, why do we even do it? Firstly, you can make plants disease resistant. So this is important because diseases in plants can cause, in the worst case scenario, famines. Probably the most famous one being the Irish potato famine that was caused by a fungus pathogen called Phytophthora, which means plant destroyer.
Yeah.
And that obviously led to the deaths of millions of people. So if we can prevent that, why wouldn't we? Especially with the growing population and shrinking land available for agriculture, we need to have all the help we can get, basically, in making these food crops as good as they can be. So disease resistance is a huge one. Another reason to gene edit plants, and by the way, when I'm saying gene editing, this creates what's called genetically modified organisms, GMOs. Technically, that can apply to any organism that's been gene edited, but most of the time in day-to-day conversation, when we're talking about GMOs, we mean gene edited crops, because in certain countries like the US, that's usually what you're eating.
Yeah, but in Europe,
in the European Union, they're illegal. So it does depend on where you are.
And it's still, that's just as the UK has taken on all of the EU's law to begin with, it's still illegal here. That could change.
That could change, and fingers crossed it does. But
and then gene editing in animals, I'm guessing, is at an earlier stage of development.
It is in an earlier stage of development. We'll get to some of the applications for CRISPR to animals in a minute, but it has been done, but it's not widespread.
Right.
Yeah. So we had disease resistance. You can also induce in plants herbicide resistance, and the benefit of that to a farmer is you can't have weeds competing with your crops, and so but you can't individually go through and hand pull all the weeds. That would take forever. You'd run out of time. So what farmers like to do is to spray their whole field, but if you spray your whole field with normal crops, you're going to damage your crops. Yeah. So if you can, be growing crops that are resistant to your herbicides, then you can spray the whole field. It'll kill the weeds and leave the crops alone, and it'll be fine.
Cool.
And you can also have pest-resistant crops. So typically the way that works is they'll insert a gene from bacteria that creates a toxin for insects. So when the insects eat the crop, they die.
Right. But these toxins aren't harmful to humans,
that's a really great question. They're not harmful to humans or mammals of any kind. They've been shown to have detrimental effects to like butterflies, which obviously are a lot more closely related to the average pest. But butterflies don't normally consume the pollen of crops, so it's not usually an issue.
Right, and these things can be adjusted over time. And
of course, things can change. But right now they're only toxic to insects, which is what they're supposed to be toxic to. So it's not an issue. And then there was one more, nutrient enrichment is another reason that you might gene edit crops. And the most famous example of this is golden rice, which started off as an initiative by the Rockefeller Foundation in the early 80s. But took some time to develop, but now it's fully developed. And the problem they were trying to solve is vitamin A deficiency in children, which can cause which on an annual basis was causing 670,000 deaths in children, Jesus. Particularly young children, and 500,000 more cases of permanent blindness. So these kids were going blind because they didn't have enough vitamin A and staying blind for their whole lives. And where this particular problem is in places like Asia, where the children mainly have rice as their only form of food. They didn't really have a lot of access to fresh fruit and vegetables that could provide those vitamins. So the solution was, we'll swap out the kind of rice they're growing and create this vitamin A enriched rice that happened to be kind of like yellow golden in color. That's why they call it golden rice. And it has made huge strides towards solving the problem and reducing the number of child deaths from this deficiency. So those are the applications of gene editing in plants generally.
That's a lot of really significant help for really especially poorer people in
almost definitely. And as I said, growing population, shrinking agricultural land, we need all the help we can get trying to feed a population.
Most of that population growth is in poorer countries as well. There's places like India, Africa that have the exploding populations where they're already struggling for resources. So
exactly. So this technology is hugely helpful to them. And so how CRISPR is being done, or sorry, how gene editing is being done, particularly in the last decade, is using CRISPR technology.
Right.
So
this is what we've had the new development in.
Precisely, yes. But CRISPR was actually discovered by Jennifer Doudna, I hope I'm saying that right, and Emmanuel Charpentier. They discovered this CRISPR technology and got the Nobel Prize for it in chemistry in 2020. But they discovered it in like 2012, 2013 time. And basically, let me read what CRISPR stands for here, if I can find it, sorry. So this technology, the discovery was found in bacteria and how they fight viruses. So CRISPR stands for clustered regularly interspaced short palindromic repeats. What?
So palindromic is strange to me because that word is, it means a word where the letters are the same whichever way around you read it. So,
well, I think probably part of the reason for the name is if you think about DNA, it's always represented in letters, and there's a lot of repeats.
Right.
Because of the letters come from the shortened nucleotides that we were talking about last week. It's just the first letter of the nucleotides. But what it actually is bacteria keep copies of virus RNA as kind of mugshots so that when a virus attacks them, they can recognize the virus and use an enzyme called Cas9 to remove it. And so scientists have figured out how to replicate this RNA, this CRISPR, to identify the gene that they're interested in, and then they started out just using Cas9 to deactivate it or snip it out.
Right.
So the first applications of this technology were to deactivate a gene or to remove an undesirable gene. And what genes might you want to remove? Those would be the kinds that, for example, make you susceptible to in the case of a plant, susceptible to a disease. For example, just a really basic application.
Right.
And they've moved on to be able to do more than that. And now they can replace a nucleotide, like we were talking about last week, where you can have one nucleotide swapped.
So this, so sort of the like mutations that cause cancers could be corrected theoretically.
In theory, yes, and they have done this in animals. So this is one of those.
Right.
What I was saying.
Not cured cancer in animals,
not cancer, but genetic
defects,
disorders using this technology. They've cured genetic disorders. They've sterilized mosquitoes. They've prepared pig organs for transplant into humans. They're just starting to test it on humans now. It's sort of people have a lot of safety concerns, which are to some extent understandable. It's a new technology.
Yeah, it needs a demonstration period. Yeah.
But so far, it's been perfectly fine. It's actually relatively cheap and easy to use technology. So highly trained scientists are usually fine to do it. But yeah, so those are some of the applications of it, and it works by, as I said, deactivating, or now they can activate a gene, because you have a lot of genes in you. Not all of them are being expressed, and by being expressed, I mean your genes are used to create amino acids that are used to create proteins, but your genes aren't all doing that all the time.
Yeah.
That would be insane.
It's like implementing a computer program almost. Yeah.
Yeah. So, but using this technology, they can get a gene that wasn't being expressed to express itself nowadays. So there are a lot of cool applications of CRISPR, and the technology has advanced since it was first introduced 9 years ago. And the most recent, so this is the new discovery, the most recent advancement in this CRISPR technology is called CRISPR Act 3.0. And it was developed by a team led by Yipyong Chi. And basically, the advancement that his team was able to develop is that you can activate or boost 7 genes at a time. So this technology, compared to earlier CRISPR technology, it has 4 to 6 times the gene activation power of the earlier versions. And this has been tested so far, just in plants. They started with rockcress, which is Arabidopsis, which is plant geneticist's favorite plant to use for this kind of thing.
Why?
It was the first plant to have its genome sequenced.
Is it sort of particularly easy to?
It seems to be very easy to work with. I'm not sure genetically why that is, but it does breed really easily. and quickly,
so
so
it's
you
the
can.
speed that you can watch changes happen at. Yeah.
But to make sure it's applicable to crops, this team also tested it on tomatoes and rice, and
Right.
had no problems. One of the tests they performed was getting the plants to flower faster. So that would be boosting the genes for flowering and getting them to move more quickly, which would be helpful for things like speeding up breeding programs. If you can get them to flower faster, then you can get the next generation faster.
Right. So you can sort of breed and develop the crop more quickly. Yeah.
Yes, exactly. So that, again, just the applications of that for plants, we've got disease resistance, pest resistance, herbicide resistance, nutrients. There's all kinds of fantastic applications that we could see from this technology. So it's really, really helpful to be able to be moving that forward.
Awesome.
I think that's all I had on that. So if you wanted to kick off our honorable mentions for today.
Sure, yes. And the first one of those I wanted to talk about was the discovery of what they're calling the galactic arc, which is a bit of a misnomer, really, because this is a 3.3 billion light year long structure in space. And
so it's not an arc.
No, it is an arc.
Okay.
It's just too big, I think, to call galactic.
Okay.
Because to give a sense of scale, so a light year is the distance that light can travel in 1 year. And to give a sense of scale, the sun is 8 light minutes away from Earth. And then the nearest star system to us, the Alpha Centauri system, is 4 light years. Our galaxy is 100,000 light years wide. The next galaxy to us, Andromeda, the next large galaxy to us, is 2 million light years away. And that's 100,000 light year wide galaxy, nearest two galaxies, 2 million. This is 3.3 billion. So galaxies form into clusters, those clusters form into superclusters. This is an arc of superclusters. So this is ridiculously large structure. This is by far the largest structure that's ever been detected. And the reason that's significant is because the cosmological model suggests that, which is the model that astronomers have been using to understand the structure of the universe, is a mathematical model that predicts that at a certain level of scale, the universe should become even. There's a word that I've sort of forgotten for a moment, which is uniform. Thank you. So at any level of scale, start us, we have structure at this scale. You and I are separate beings. We have form. There's space between us. All of this stuff has structure. The planet has structure. The solar system has structure. The galaxy has structure. And these clusters of galaxies have structures. They move around each other. They orbit. There's dense regions and non-dense regions and filaments of galactic clusters and spaces between them. But the model predicted that when you get up to the sort of billions of light year scale, and this 3.3 billion light years is 7% of the estimated total size of the universe. So it predicts that at this point, everything should even out and it should just form into a sort of a haze where everything's spread out evenly. There's no real differentiation between different regions. And instead, you've got this whopping great arc with space around it.
That doesn't fit that model at all.
No. So it's saying that there is structure at this largest scale, which means that there's something wrong with the model. Now, when you're observing structures of this kind of size, you can make errors in observation.
Of course.
But they're saying that there's a 0.003% chance of an error in determining this, which is difficult because the further away something is in space, the longer it takes light to reach us from it. So if something's 3 billion light years away, it's going to take 3 billion years for that light to reach us. So the more distant parts of the arc are older than the earlier parts. So you have to make all these calculations to correct for that. But no, it's almost certain that this is a structure, which means that, again, like we were saying before with the new species of human, if the evidence is telling you that your model's wrong, it's your model that's got to change most of the time.
Makes sense to me. That's kind of crazy, though. That's out there. The next thing that I wanted to just briefly mention is the concept of nuclear batteries. So this is a proposal that's been put forward by a group. I'm going to read their names so I don't forget anybody. We've got Jacopo Bongiorno, Robert Frieda, Stephen Auermeyer, and Kevin Chilton have put together a proposal based on their research with nuclear power. And the idea that they've put forward is a way to have essentially what they like to call nuclear batteries. It's a miniature nuclear fission plant, essentially.
So this is existing nuclear technology, but on a smaller scale.
On a smaller scale. And rather than the concrete that you'd want normally around a nuclear structure. You have steel shields. But the idea is you assemble it in a factory and then you would deliver it to the customer, who might be, a factory that's building things, or a military base, or ideally, eventually, even a residential neighborhood.
Yeah.
You plug this battery in to the grid in that area, and it would power it for 5 to 10 years without needing any maintenance or refueling or anything. At the end of that time period, you take it away for refurbishment and replace it.
Right.
And of course, as we were saying last week, the advantage of nuclear technology is zero carbon emissions. You do have to deal with the waste product, but it's very environmentally friendly and very reliable.
Yeah.
And an advantage to this particular idea or model is that you're not really going to have an explosion or a fire or anything like that because it's so small. So firstly, it's not generating that much heat just because it is that small. And secondly, because it's so small, it's got a large surface area to volume ratio, so it can passively cool itself.
Right. And if it's not producing much heat, it's probably not producing as much radiation. Period heat is a form of radiation. So
I'm quite right.
Even if something did go horribly wrong and there were a leak, it would be a much smaller scale problem than.
And the authors of this proposal also suggest installing it underground, which would both limit the amount of radiation that could get out if there was some kind of accident, and it would also make it less vulnerable to attacks by terrorists or what have you.
But it's worth noting, I'll tell a little story quickly, which is that in the 1980s, British Rail were starting to carry nuclear waste in flasks by train, and there was a lot of public outcry about, we don't want radioactive waste coming through our towns. So they staged a crash test and they put one of these flasks across a railway line and drove a 300-ton train into it at 90 miles an hour, and the thing was completely undamaged.
Right. So when it's done correctly, there's no reason to be worried about radioactivity getting out. And the one of the authors of this paper did an interview where he was saying, this isn't ready for market yet because it hasn't gone through that testing.
Right.
So that would need to happen first, but there's no reason that couldn't happen in the next couple of years. And the idea is to produce one of these would take only a few weeks. So there's no reason that couldn't be done. And moreover, NASA is testing a similar concept. So they have used nuclear power before, but they use natural radioactive decay to power their deep space probes.
Right. Yeah.
RTGs, I forget what exactly that stands for, but radioisotope thermogenerators, I think.
Yeah. So because those further away from the sun you are, solar power doesn't work.
Right.
But I know the Voyager probes they've been running for 50 some years now. It's 1977, and they're still running on those.
Right, and those use plutonium, but NASA is now testing. They call it a microreactor, but it's a similar concept to this nuclear battery idea. They've developed it within 3 years. They want to use it to power their manned mission to the moon. They're planning for 2024. So a lot of potential there in making more reliable, clean energy that's safe, that we could all have in a number of years.
Hopefully it'll get used on a private mission to the moon as well at some point.
That'd be exciting.
Sort of on that note, the next thing I wanted to talk about is the Federal Aviation Mission, the FAA in the US, has granted the first ever license for commercial space tourism to Virgin Galactic. So Virgin again, we last week talked about Virgin's air taxi plans, and this week it's Virgin Galactic who are going to operate a spacecraft called Spaceship Two. Spaceship One was the first ever private vehicle to reach space back in 2004, built by a company called Scale Composites. A man called Burt Rutan's private dream to build this company and develop aviation technology. And
I don't want to put you off your flow, but I was just thinking, what is the scale of the vision of Richard Branson and his company?
Well,
is enormous.
It is. I was just watching a video about his Hyperloop plans last night as well, but and he's done so much, but he is a visionary and he has a lot of money behind him, but he couldn't do what he does without people like Burt Rutan who develop the technologies that he then puts his money into to bring them to where they're at. So Virgin's money helped Scale Composites develop White Knight 2 and Spaceship 2. So White Knight 2 is a plane with two twin fuselages, and then Spaceship 2 sits between the fuselages under the wing, and it flies up to 50,000 feet, I think, maybe to higher, and then Spaceship 2 drops out from under the aircraft, fires its rocket motor, and goes up to the edge of space.
And they've done a test flight of this, haven't they?
They've done many. So Spaceship One operated on the same principle, and Spaceship Two has done a number of test flights above the edge of space. So I was going to say this is a suborbital spacecraft, the same as if you watch our first episode, New Shepard, which is built by Jeff Bezos' company Blue Origin, is also suborbital, but that's a rocket that takes off from the ground. This is an aircraft that is deployed by another aircraft.
Sure.
So it's a more efficient way of getting to the edge of space. As I say, this doesn't go into orbit, which requires a lot more energy to reach the speed you have to reach to stay in orbit around the Earth. This just is designed purely for tourism, really. It just goes to the edge of space. You get the weightlessness, you get the view of the Earth from space, and then you come back down, and it doesn't go all the way into orbit. But it's a very interesting vehicle. The wings rotate round in space, and it comes back down like a shuttlecock, like a badminton shuttlecock. Sort of floats back into the atmosphere. It's a very clever piece of engineering.
And you can see this in the test video that we'll link to.
Yeah, so there's a great little video that Virgin's released. Richard Branson plans to fly on the first flight, so he's up against Jeff Bezos, who's planning to do the same thing. So we have two entrepreneurs racing each other. This is the best kind of space race, racing each other to ride their own spacecraft to orbit, to space for the first time. And that's much more exciting to me than
excellent competition.
than NASA versus the Chinese, which is I'd much rather people like these guys were taking us to Mars as well. In the race of NASA versus the Chinese to Mars, I think Elon Musk might win that race, but we'll see what happens there.
Yeah.
But
the potential of this flight, and to a lesser extent Blue Origin's plan too, is that eventually, it starts out the ultra wealthy are the only ones who can afford it, but it'll get to moderately wealthy people.
And I think that's one advantage of Spaceship Two's way of operating, is that it's lower cost than the traditional rocket. So I think the cost of that will come down quicker and sooner. And I think that's for purely for the tourism side, that's a much more realistic way for regular people to get access to space quicker than an orbital system or a rocket-based system is. And I think people probably feel more comfortable getting on a plane than on a rocket.
Probably.
Even though it's still a rocket at the end of the day. There's one other thing I wanted to mention quickly, which is that Virgin have experience using systems like this already, because they have another company called Virgin Orbital, and that uses a system called Pegasus, where they have a former Virgin Atlantic Boeing 747 that flies up to normal cruising altitude, and then it has a rocket under the wing which carries satellites into orbit. So that's more of a traditional sort of missile style rocket, but it's launching from an aircraft rather than from the ground, so it has a lot less atmosphere to get through, and it can just basically go up and reach orbit more easily. So it's the same principle really, and they already using that to launch very cheaply, launch small payloads into orbit. So that's often a way that smaller companies can get satellites into space. So
payload would be like a satellite or something. Okay.
Yeah, whatever it's carrying, but pretty much always satellites in this case, or for one purpose or another.
Sure. Cool.
So Virgin's doing so much. It really is incredible the amount of things that Richard Branson gets his fingers into.
Exciting. I think the last thing I had for today was a group of researchers at the University of Maryland, led by Zhangwei Li, have managed to develop. They're called organoids, but they're basically synthetic parts of a kidney.
Right. Okay.
Because they're not the full organ. So
because organoid does sound like a Doctor Who villain.
It does a little bit, but it really just means part of an organ.
Right.
And in this particular case, they've managed to get this synthetic tissue that they've grown from stem cells in the lab to mimic one of the key functions of the kidney, which is to concentrate and transport urine.
Right.
So stem cells, of course, are cells that haven't decided what they're going to be yet, basically. They're undifferentiated is the proper term. But they can be made to be a number of different things. So they're being used for a lot of kinds of really exciting research and treating a number of disorders, diseases. In this case, obviously, the goal would be to treat genetic disorders that affect the kidney and kidney failure.
Yeah, my dad had renal failure when I was 8 years old, so I saw how debilitating that can be.
Exactly. So it's a huge step forward. So far, they've already used it to mimic a genetic disorder just for study, and they want to use it to screen potential treatments before they try it on an actual person. And then it's a step toward being able to develop a fully synthetic kidney that could, be transplanted into somebody who's in an advanced stage of kidney failure. So huge potential for improving and saving lives there as well.
Something I'd very much like to see.
Excellent. So I think that's all we have for today.
Cool.
So today we talked about.
So I talked about the discovery of Nesher Ramla Homo, the new species of prehistoric human.
And I talked about the CRISPR Act 3.0 advancement in gene editing in plants.
And then in our honorable mentions, I started off with the discovery of the galactic arc and what that means for our understanding of cosmology.
And I continued with a discussion of nuclear batteries and the proposal that's been put forward to use those.
And then I went on to Virgin Galactic's awarding of a license by the FAA to fly tourists to the edge of space.
And I wrapped up by talking about the researchers who developed parts of a synthetic kidney.
Wonderful.
So thank you for joining us on Innovation Celebration.